Golden blood. Start with the golden blood itself, because it sets the scale for everything else. Rh-null was first documented in 1961 in an Aboriginal Australian woman whose blood baffled clinicians because it reacted to nothing in their standard antigen panels. The mechanism is a mutation in the RHA gene, which normally acts as a scaffold holding the Rh antigen proteins onto the surface of red blood cells. Break that scaffold, and every Rh antigen simply fails to anchor, leaving blood that's a blank slate immunologically. Here's the catch nobody advertises. Golden blood can be given to almost anyone, but almost nobody can give blood back to a golden blood carrier. So these individuals often travel internationally with documentation of their own status in case of emergency, because a compatible donor might be a continent away. HIV immunity, CCR5. Immunity works the same way, just against a virus instead of another person's blood. Roughly 1% of people of Northern European descent carry 2 copies of a mutation called CCR5 delta 32, identified through work led by geneticist Stephen O'Brien at the National Cancer Institute in the 1990s. The mutation deletes 32 base pairs from the gene that builds the CCR5 receptor, which is the exact docking point HIV uses to enter white blood cells. No receptor, no entry point, and carriers turn out to be almost completely resistant to the most common strains of HIV. Here's where it gets interesting. Researchers believe the mutation was originally selected for because it protected against an entirely different disease centuries ago, possibly smallpox, meaning a defense built by medieval immune systems is still running interference against a virus that didn't exist until the 1980s. Supertasters taste runs on the same logic of small deletions producing large effects. About 25% of people are supertasters, a term coined by Yale researcher Linda Bartoshuk after she noticed wildly different reactions to bitter test compounds among her subjects. The gene involved, TAS2R38, controls bitter receptors, and supertasters simply carry more fungiform papillae, the mushroom-shaped taste bud clusters on the tongue, than everyone else. The effect isn't subtle. Supertasters statistically consume less fat, less sugar, and less alcohol across their lifetime, because everything registers more intensely, meaning one gene variant quietly shapes decades of dietary habit without the person ever realizing why broccoli tastes like punishment to them, specifically. Lactase persistence. Digestion tells almost the opposite story, one of a trait most humans lost rather than gained. Every mammal produces lactase as an infant to digest mother's milk, and every mammal is supposed to stop producing it after weaning. Roughly 7,000 years ago, in populations across northern Europe and pockets of East Africa, a mutation near the LCT gene kept lactase production switched on into adulthood, a trait geneticists call lactase persistence. A 2007 study led by anthropologist Sarah Tishkoff at the University of Pennsylvania traced multiple independent origins of this mutation across different continents, meaning evolution arrived at the same solution more than once in populations that never had contact with each other. Here's the thing. Globally, lactase persistence is actually the minority trait. Roughly 65% of the world's adult population loses the ability to fully digest lactose, which means the normal most Western countries assume is actually the genetic exception. No body odor. ABCC11. Smell has its own quiet mutation, and this one shows up on nobody's medical chart because it isn't a disorder, it's an absence. A variant in the ABCC11 gene, especially common across East Asian populations at frequencies above 80%, changes the composition of a person's sweat and earwax at the same time, producing dry earwax and, more relevantly, virtually no underarm body odor, because the specific compounds bacteria feed on to create odor simply aren't present in the sweat to begin with. Researchers at Tokyo's National Institute of Genetics confirmed the same single gene controls both traits, meaning a doctor could theoretically predict whether someone needs deodorant just by checking their earwax texture. The athlete gene, ACTN3. Athletic performance has its own named variant too. The ACTN3 gene codes for a protein called alpha-actinin 3, found specifically in fast-twitch muscle fibers. The fibers responsible for explosive, short-burst power like sprinting or weightlifting. a version of this gene, identified through research led by geneticist Catherine Norris, because its presence correlates strongly with elite sprint and power athletes, while its absence is overrepresented among endurance runners. Here's the mechanism in plain terms. Without functional alpha-actinin 3, muscle fibers shift slightly toward more efficient, fatigue-resistant function, trading raw explosive power for stamina, an actual measurable trade-off sitting inside a single gene. Hyperthymesia memory produces one of the strangest documented cases in modern neuroscience. In 2000, a woman named Jill Price contacted neurobiologist James McGaugh at UC Irvine, claiming she could recall nearly every day of her life since age 14 in diary-level detail. McGaugh spent years verifying her memories against historical records before publishing on what's now called hyperthymesia, or highly superior autobiographical memory, confirmed in fewer than 100 people worldwide. Brain scans reveal enlarged temporal lobe structures and denser connections in memory circuitry. But the twist is this isn't linked to general intelligence or better memorization of facts like phone numbers. It's hyperspecific to lived personal time. Their brains aren't better at remembering. They're structurally unable to forget their own life. The photic sneeze reflex. Sneezing has its own bizarre genetic quirk, hiding in plain sight. Somewhere between 18 and 35% of people experience the photic sneeze reflex, an involuntary sneeze triggered by sudden exposure to bright light, sometimes nicknamed a CHU syndrome by researchers, an actual acronym for autosomal dominant compelling helio-ophthalmic outburst. The mechanism is thought to involve a crossed wire in the trigeminal nerve, which carries both light signals and nasal irritation signals close enough together that the brain occasionally misfires one for the other. Here's what nobody expects. This trait is autosomal dominant, meaning if one parent has it, each child has roughly a 50% chance of inheriting it. And fighter pilots have historically been screened for it, because an uncontrollable sneeze at the wrong altitude is a genuine safety risk. Heterochromia. Eye color carries its own visible signature of incomplete genetic instructions. Heterochromia, where a person has two different colored eyes or multiple colors within a single iris, results from uneven distribution of melanin, usually due to a mutation, injury, or a mosaic genetic pattern established early in embryonic development. Most cases are entirely benign, though ophthalmologists sometimes screen for underlying conditions like Waardenburg syndrome, first described by Dutch ophthalmologist Petrus Johannes Waardenburg in 1951, when it presents alongside hearing loss. Sound familiar? Because it's the same story as golden blood and dry earwax. A scaffolding protein or pigment cell simply doesn't distribute evenly, and the visible result looks dramatic, despite the underlying cause being a single misfired signal during early development. Cilantro tastes like soap, OR6A2. Taste strikes again, with the most viral genetic quirk of the last decade. Cilantro tasting like soap to a specific slice of the population comes down to a variant near the OR6A2 gene, part of a receptor cluster tuned to aldehyde compounds, the same chemical family present in soaps and lotions. The 2012 genome-wide study through 23andMe, led by researcher Nicholas Erickson, scanned over 14,000 genomes and confirmed the link, finding the soap taste reaction in between 4 and 21% of people, depending on ancestry. This isn't a matter of an undeveloped palate. Their receptors are reading the exact same molecule as everyone else and translating it into an entirely different signal. The longevity gene, FOXO3. Aging has a genetic outlier too, one still being actively studied for its implications. The FOXO3 gene, involved in regulating cell stress response and repair, has a specific variant strongly overrepresented in centenarian populations. A study led by geneticist Bradley Wilcox, examining Japanese-American men in Hawaii as part of the long-running Kuakini Honolulu Heart Program, found this FOXO3 variant appeared significantly more often in men who lived past 100 than in the general population. The mechanism ties into how efficiently cells manage oxidative stress and initiate repair before damage accumulates into disease, meaning some people are quite literally carrying a better maintenance manual for their own cells. Double eyelashes, FOXC2. Even eyelashes have a documented genetic anomaly attached to a real historical figure. Distichiasis, the growth of a second extra row of eyelashes, is linked to mutations in the FOXC2 gene, and was famously associated with Elizabeth Taylor, whose double lashes were widely reported and occasionally misattributed as purely cosmetic rather than genetic. In most documented cases, Distichiasis is linked to a broader condition called lymphedema distichiasis syndrome, involving swelling caused by lymphatic system dysfunction, though isolated double lash growth without the syndrome does occur. Here's the pivot. A trait admired as an aesthetic feature in magazines is, mechanistically, the exact same category of event as every other trait on this list. A gene issuing an instruction one extra time when it was only supposed to fire once. Synesthesia. Perception itself can be rewired. Synesthesia, where stimulation of one sense involuntarily triggers a second, such as seeing specific colors when hearing specific sounds or perceiving numbers as inherently colored, affects an estimated 4% of the population, according to research compiled by neuroscientist Julia Simner at the University of Sussex. Brain imaging shows increased structural connectivity between sensory regions that are normally more separated, meaning the wiring between, say, the auditory cortex and visual processing regions is denser than average. Composer and musician Pharrell Williams has spoken publicly about experiencing sound as color, describing songs in terms of their hue rather than only their pitch, a documented modern example of a trait that's been recorded in written accounts for over 2 centuries. Red hair and pain resistance, MC1R. Pigmentation and pain sensitivity turn out to be linked through the exact same gene. Red hair, found in roughly 1 to 2% of the global population and up to 13% of people in Scotland, comes from mutations in the MC1R gene, which normally directs pigment cells to produce dark eumelanin, but instead produces reddish pheomelanin when mutated. A 2021 study out of Massachusetts General Hospital, led by researcher David Fisher, found that this same MC1R disruption also affects opioid receptor pathways in the brain, meaning redheads require meaningfully higher doses of certain anesthetics to achieve the same effect as everyone else, while also showing increased sensitivity to certain types of pain like cold and pressure. One pigment gene, and it changes both what color grows out of the scalp and how an entire nervous system negotiates with pain. Malaria resistance, sickle cell trait. And then there's the trait that reads like evolution actively trading one danger for another. Sickle cell trait, carried by roughly 8% of Black Americans and up to 40% of some populations in parts of West Africa, comes from a single mutation in the HBB gene, altering hemoglobin just enough that some red blood cells distort into a crescent shape under low oxygen conditions. Carrying one copy causes no significant disease in most people, but it does something remarkable. It makes the carrier's blood cells substantially harder for the malaria parasite to survive inside of. A defense first proposed by British geneticist Anthony Allison in the 1950s, after he noticed the trait clustered geographically almost exactly on top of malaria endemic regions. Its evolution making an explicit trade, tolerating a mild, usually invisible blood quirk in exchange for a shield against one of the deadliest diseases in human history.